Hi, welcome to our lecture on the Introduction to the Nervous System. In this chapter, we're going to introduce some basic nervous system terms, as well as take a look at some of the basic rather than specific parts of the nervous system. We're going to begin here with this idea that we can divide the nervous system up into two different basic anatomical divisions. And these are going to be the CNS, or central nervous system, which consists of the brain and the spinal cord. That is, the part of the nervous system which resides within the axial skeleton. And then we have the peripheral nervous system. The peripheral nervous system is going to be this portion which is going to extend out from either the brain or the spinal cord, meaning that it can carry motor commands out to other parts of the body or receive sensory information from other parts of the body and bring them back to the central nervous system for processing. This portion of the nervous system is going to consist of cranial nerves coming off the brain, spinal nerves coming off the spinal cord, and ganglia, which we're later going to learn are clusters of neuron cell bodies found in the peripheral nervous system. So when we think about this PNS, since there are signals going in a couple of different directions, we want to divide that up and understand how this is happening. And we do have the afferent and efferent divisions of this. The afferent, or more correctly pronounced afferent, is going to be into. That is, it's going to bring sensory information from the periphery of the body back to the central nervous system. And the efferent, or efferent division, is going to take information away from the central nervous system. So it's motor information being taken out to what we call effectors. It's nice that we can link effectors and efferent here. It is important to recognize these are afferent and efferent, but if we say them in that way rather than afferent and efferent, sometimes there is some confusion, so people will commonly pronounce them with an A and an E sound, simply for clarity. Now, if we take a look here at how this kind of works together, we have an actual reflex arc, which means we're going to ignore the brain at this point. We're going to receive sensory information from the skin. We have afferent transmission coming back on a sensory neuron to the posterior horn of the spinal cord. It gives that information to an interneuron. These are neurons that reside entirely within the central nervous system. That is, in this reflex, going to carry that information to a motor neuron. It has a cell body residing in the anterior horn of the spinal cord. And the axon of this neuron will take that signal out here to an effector, in this case skeletal muscle. So if we think about the basic kinds of effectors that we have in the body, it's pretty obvious. Effectors are things that do stuff, and there aren't that many things in the body that really do things. We have muscles. It can be skeletal muscle, smooth muscle, or cardiac muscle. It doesn't really matter which, and we're going to have those be able to contract, which of course we talked about muscles being able to do. And then we could have our effector be a gland. You can actually see a neuron down here in the bottom picture carrying information out to a gland. And glands, of course, also do something. In their case, they secrete. So if we kind of look at what you really are, there are a couple things you can do. You can contract, you can secrete. There's not really much else that you're capable of. Now, of course, we kind of can use those things for a variety of purposes. So when we think about where neurons are going to go, they might go to another neuron, but they could go to either of our kinds of effectors. All right, we do have different signal types within the afferent division and within the efferent division as well. Within the afferent division, those sensory signals can be either somatic or visceral. Somatic signals, coming from the root word soma, meaning body, is information we're going to be conscious of. Primarily, this is the cutaneous senses, and we're bringing back that information from the skin, such as touch and temperature. And then for the opposite of this, the visceral, that's going to be kind of information that is important, but that we are unaware of. So when we have receptors that are getting information regarding our blood pressure or our blood chemistry, such as the concentration of oxygen or carbon dioxide, that information isn't something we are aware of in a conscious state. But we do have, of course, a portion of our nervous system which can respond to any imbalances we detect and fix those. So we still do have some very important information here. Now, as far as this diagram down here, we can tell that both the somatic and visceral sensory information come into the posterior horn of the spinal cord, so they are going to roughly the same location. Now we do also have what we call our special senses. And special senses are somatic, but we do think about them as being a little bit differently. These are the kind of sensors that are located in our head and are giving us information like smell and vision and hearing. And these, of course, are things where we are conscious of the message that we receive. It is important to recognize that there actually is going to be some kind of subconscious level processing of this information as well. For example, we have what we call the corpora quadrigemina on the posterior side of our midbrain, which will both transfer the information regarding hearing to the parts of the brain that will consciously perceive that, but also subconsciously deal with that hearing information to help us create spatial maps that relate the space around us to things like sound and sight as well. Now, in the efferent division, we again have somatic, so we're using that word repeatedly. That's probably important. We want to learn this word. We also had somatic cells, which happen to be the general body cells. Somatic efferent signals are going to be the voluntary ones. They're the ones that go to skeletal muscles. And then the opposite of this would be autonomic. These are the involuntary signals that go to smooth muscle, to cardiac muscle, and to glands. So if we think about this, if our sensory information that was somatic was stuff we are conscious of, we have a very kind of equivalent version of somatic here where it is stuff that we are aware that we are doing. We are intentionally trying to do something that is a somatic signal. Whereas the autonomic signals tend to be done in response to visceral sensory information. And when we use something like smooth muscle or cardiac muscle or glands, in this case we are basically doing something, but we're not doing it with the intent to do it. It just kind of happens. Now that doesn't mean that we won't be aware of it afterwards. So for instance, if I get hot and then I sweat, the getting hot is going to be a visceral sensory information. That visceral sensory information is going to be responded to through an autonomic response. I'm probably going to use some smooth muscle so that I can actually provide more blood to my skin, but I'm also going to secrete from my sweat glands. I'm of course going to feel that I'm sweating, and that's going to be through that somatic sensory information that I'm receiving from cutaneous receptors in my skin. So there will kind of always be these constant relationships between these different divisions. Next thing we're going to take a look at is the different parts of the neuron. Now we're just going to be taking a basic look at a neuron, and you'll look at these more closely in physiology class. We want to be aware that they have dendrites, a cell body, and an axon. So the dendrites occur in multiples. They are highly branched. They are often going to be receiving information either directly from the environment or through a synapse with another neuron. The soma or cell body contains the nucleus, contains mitochondria, it contains ribosomes, most of the organelles. It is important to recognize that there will be organelles in other portions of this because we do have a need for mitochondria and the ATP they provide along the length of the neuron. The axon then is this single component here which extends away from the cell body, at least in an example neuron like this, which looks roughly like a motor neuron or like some interneurons. And this will occur singly. It's going to conduct nerve impulses towards what we call a synaptic terminal, that is to another neuron or to an effector. And then we're going to communicate with another cell over what we refer to as a synapse. That means that axons can, in some cases, be incredibly long, going, let's say, all the way from the lumbar region of your spine down to the plantar surface of your foot. So what exactly is a synapse? We've mentioned that we're carrying this information to a synapse. We're going to talk specifically here about a chemical synapse, but there are also electrical synapses, which are going to consist of gap junctions and allow the flow of ions across them, as we talked about for those intercalated discs in cardiac muscle. In the case of a chemical synapse, what happens is we have a neuron bringing information, a signal, to the end of that axon, what we call the axon terminal or the synaptic knob. And here we're going to produce neurotransmitters, which are released into this cleft, this gap, in between the neuron that carried this initial signal and the neuron or effector we're trying to communicate with. That, of course, will have receptors here which the neurotransmitters can bind to. And these have the potential of either stimulating some kind of response in that postsynaptic cell or inhibiting that response. Therefore, we can actually have an interaction between inhibiting neurotransmitters and stimulating neurotransmitters here, which determine whether or not the postsynaptic cell does whatever it's going to do. If it's a neuron, go ahead and have another nervous system signal, or if it's an effector, do whatever that effector does, contract or secrete. Now, we do want to recognize we do have different terms which we use to talk about different kinds of neurons. So if we think about in that afferent division, we had those sensory neurons. Well, sensory neurons carry information to the central nervous system, that is sensory information. We then have motor neurons in the efferent division, which carry information from the central nervous system to effectors, and they cause them to do something. We think about this as being motor because we generally think about the control of our musculature as being motor signals. Of course, this could be information, if it is autonomic in nature, that goes to things other than skeletal muscles. It could even go to glands. And then we have interneurons, which are going to be located entirely in the central nervous system. So we see a sensory neuron bringing information back to the CNS. You might notice that its cell body is in a weird position relative to the other neurons we're taking a look at. So we have dendrites in the skin. An axon carries information and bypasses the cell body to get to the end of that axon. It delivers information to the dendrites of this interneuron. We can see its cell body here, and it has a short axon extending out and then branching to interact with the dendrites of this motor neuron, which also has a cell body near its dendrites, and in this case, a long axon that goes out to an effector. We also have neuroglia within our nervous system. We're going to recognize 4 types of neuroglia, or glial cells, within the central nervous system, and 2 within the peripheral nervous system. Now, there are some similarities between the jobs that they take on, but we're going to begin here with, again, the central nervous system with astrocytes. And we can find an astrocyte in here. You'll see that it's connected on one end to a capillary, wrapping that capillary up, and on another end to a neuron. So these are going to help to control the interstitial environment within the central nervous system for the neurons that are in there, producing a portion at least of what we refer to as the blood-brain barrier, which keeps the chemical signals, hormones, and things like that floating through our bloodstream from interacting with our nervous tissue and causing it to get all wonky. Now, the ependymal cells, which are our next type, are also sort of contributing to that blood-brain barrier. We have ependymal cells here next to the central canal of the spinal cord, and we're going to have cerebrospinal fluid, or CSF, flowing through that central canal. These ependymal cells are going to be involved in secreting, circulating, and monitoring the cerebrospinal fluid. Now, this cerebrospinal fluid is the fluid which our brain floats in, and obviously, being that fluid environment, it's going to be involved in the exchange of nutrients and wastes with the nervous tissue. So again, we want to make sure that when these ependymal cells, the ones that are secreting CSF, are associated with blood capillaries and are producing this fluid, that they aren't putting in any chemicals that are going to confuse our brain. The other kinds that we have in the central nervous system are microglia, which act as phagocytes. We can see a microglial cell here, and it's normal to have phagocytes, cells that eat debris within any kind of tissue. These just happen to be the ones specific to our central nervous system. And then we have oligodendrocytes, which are going to have a direct counterpart in the peripheral nervous system, so we have to pay really close attention here. Oligodendrocytes are these cells where we can see their nucleus here, and they send these extensions off to these axons, and they are wrapping them up in this lipid material referred to as myelin. So they're producing what we refer to as the myelin sheath, which will help to speed up nerve transmission. And we are going to have a myelin sheath on some neurons, at least, in both the central nervous system and in the peripheral nervous system. So we're going to begin here in the peripheral nervous system with the neuroglia, which provide the myelin sheath. In this case, this is Schwann cells. You'll notice a capital S there indicating that Schwann cells are named after a person, whereas oligodendrocytes are not. So in this case, we're again providing the myelin sheath, but in the peripheral nervous system, and the setup here is very different. This is a Schwann cell, this is a Schwann cell. So the nucleus of the Schwann cell is involved in the wrapping up of the axon, whereas when we saw the oligodendrocytes, the nucleus was somewhat separate and it was extending extensions to multiple neurons to wrap them up. So different cells doing the same job. Satellite cells are going to be sort of the equivalent of astrocytes and ependymal cells working together. In this case, we're controlling the ganglion microenvironment. That's sort of my shortcut way of explaining what's going on. But we can see here we have a neuron cell body. Neuron cell bodies, when they cluster together in the peripheral nervous system, form what we call ganglia. Neuron cell bodies, by the way, are predominantly found in gray matter areas within the central nervous system. These satellite cells are around here, and if we think about the same role of astrocytes, we want to make sure that the chemical environment is appropriate for these cells. We don't want to have any inappropriate signals happening, therefore we need to control what's going on in there. Now here we have kind of a nice table that helps to kind of link central nervous system cells together and peripheral nervous system cells together. It does kind of provide a nice idea more specifically of what satellite cells are doing: regulating oxygen, regulating carbon dioxide, nutrient, and then of course the neurotransmitter levels around these neurons. So again, making sure that they are able to function properly. We do have both Schwann and oligodendrocytes. Both of those cells are going to be involved in providing the myelin sheath. Again, the myelin sheath does something pretty important. It's going to be able to speed up nerve transmission. This is something you will mostly talk about within your physiology class, but it's important to recognize that normal nerve transmission involves the flow of ions, sodium inside the axon membrane, potassium outside the axon membrane, and then the reformation of those gradients, and we refer to this as an action potential. All of that work is required for nerve transmission down the length of an axon. But where we myelinate it, it can jump from one side of the myelin sheath to the other one almost instantaneously in what we call saltatorial conduction. So we can dramatically speed up nerve transmission by not having to go through that entire process along that area that is covered up by the myelin sheath. Now you get more detail on that. I'm providing a simplification here. And again, of course, we have our microglia removing debris and pathogens, and then our astrocytes and ependymal cells doing CSF, astrocytes more directly contributing to that blood-brain barrier and kind of acting in a very similar way to what the satellite cells would do in the peripheral nervous system. So speaking of white and gray matter, which is what we really are talking about when we talk about myelination and those Schwann cells and oligodendrocytes, the tissue or the structures that dominate the white matter tissue are myelinated axons. It is the myelin in this lipid that makes this white matter. And we can actually see that down here in a nice fresh brain, how white that is. And we're going to find that in the middle of the brain in what we refer to as the cerebral medulla. Obviously the cerebrum being the large piece of brain and the medulla being the middle of it. You might notice on the spinal cord the white matter is actually on the outside and the gray is on the inside, so those are arranged differently from each other. What dominates gray matter is a little bit more complicated. Basically, it's stuff other than myelinated axons. So when we think about gray matter, we're predominantly going to think about cell bodies, which means we're going to have synapsing occurring, which is going to be the processing of information. But it can also contain unmyelinated axons and, of course, the dendrites that are generally going to be near those cell bodies, with the exception, of course, of those sensory neurons out in the peripheral nervous system. Now, we do have a few different other terms we want to get down in here so we can talk about the nervous system. We're going to begin here with bundles of axons. So we do have bundles of axons in both the peripheral and central nervous systems. In the peripheral nervous system, these are referred to as nerves. If you take a look at this nerve, you'll notice that this looks a lot like the diagram we had of muscle cells. We even have the term fascicle here for a bundle of these myelinated axons, much in the way that we used fascicle for those bundles of muscle fibers. So we also have these wrappers around here. You can see epineurium, perineurium, endoneurium, reminding us of epimesium, paramesium, and endomesium. And so this is our large nerve. As we approach the effectors, we can take these individual fascicles and have those kind of head off towards a particular area we're going to innervate. So we will have this thing kind of unwrap and fray out to make sure that it gets to all of its targets. A bundle of axons within the central nervous system is referred to as a tract. And we can kind of see that here. We have the optic nerves heading from the retina of the eye. They go through the optic canal and get to the optic chiasma or chiasm here, where they're going to have some of them cross to the other side. The tracts of axons that take this information back to areas where we're going to process it then are going to be tracts rather than nerves. The nerves are heading out in the peripheral nervous system. Once we get to where we're running through an area of solid white matter or a mix of white and gray matter, rather than in a nerve itself, we're going to be referred to as a tract. We'll actually recognize some other tracts later on. You might be aware of the corpus callosum, which is a tract. It's a bundle of myelinated axons going between the left and right sides of the brain. Then we have clusters of cell bodies, which occur in both the peripheral and central nervous systems. In the peripheral nervous system, we're going to see them as bulges. We can see one here. This is a sympathetic trunk ganglion, and this down here is going to be a posterior or dorsal root ganglion. So ganglia is the plural, ganglion is singular. These are clusters of neuron cell bodies in the peripheral nervous system. Now, we call these a nucleus if we have a cluster of neuron cell bodies in the central nervous system. So here we see some nuclei, and they are these gray matter areas that occur within the cerebral medulla, that is this middle section of this large brain here, or we see them in areas of the brain stem. So if we were to slice into this area down here, we could also see nuclei. What nuclei are going to tell us here is that rather than just being a tract carrying information on myelinated axons from one location to another, we have a potential area of processing in these locations. There's actually a cool one in this region here, which is going to basically receive motor commands coming down from the other place where we find neuron cell bodies, this cerebral cortex, this outside layer of the cerebrum. And it's going to make sure that, say, as we're walking, we swing our arms in association with the walking. So it's going to kind of mix a couple of different functions together. But we'll see that in our nuclei.